Today I wanted to talk a bit about this paper here, which has quite a bold title. And it's almost a bit heretical, I guess, maybe for our center, because we study the interactions between hosts and their microbes. But I think they raise some really great points about the diversity of the reliance of hosts on their microbes. It's not just like humans and cattle, which we rely a lot on our microbes. There's actually quite a diversity of this across the animal kingdom. And we shouldn't just look at all animals through the lens of what we know about humans and mice and cattle, etc. I'll also introduce a little bit of work I've been doing recently on a tool that can help us estimate the microbial fractions of metagenomes, which is something that I saw there wasn't really much on before, but it also it's related to this question as well about how much microbial DNA is in our samples. Cool. So did anyone actually have a chance to read this, or have they read this paper before? Okay, cool, great. Would recommend if you are working on microbes, definitely check it out. It's a really great paper. So one of the first things they do is they kind of set the stage of like, what are people talking about the interactions between microbes and their hosts? And so they go through the literature and the supplementary table and choose some papers and say, what do people talk about when they talk about microbes and their hosts? I kind of like it that they choose papers that they wrote themselves, which is very nice. They're not just kind of shooting other people, but they're also like, actually, we made these statements, right? And so that, some examples are, like all animals, butterflies also host internal communities of microorganisms. Mammal host gut microbiomes that are immensely important to the health and likely fitness of the host. Like nearly all plants and animals, humans host a large number of microorganisms. So these are very broad sweeping statements, right? Which we're kind of assuming that every animal has a really diverse microbial community. I thought I'd add one from one of my recent papers. It's a bit of an egg in the face moment, Gut functionality and health relies on a diverse community of microbes and having the entire gastrointestinal tract. So I think that's probably a bit too broad. I don't think that's really true. But again, we make these slips, right? When we write things, I think it's important for us to be very clear with the language we use, because that really kind of dictates how the field thinks about the science that we do. Their first figure, they kind of talk about a bit of a framework for like a spectrum of how hosts can rely on their microbes. So, here you might have like say some invertebrates that might not have a very rich beneficial symbiont. So these kind of yellow circles are bacteria that are beneficial. You have animals that maybe have some, but not a really dense, rich community. So that's the kind of central thing. And then at the top, you have, the example I give here is like cattle, where we know they have very rich, diverse bacterial symbionts. And they also, characterize things into different things as well. So, there's also pathogens and parasites that inhabit animals, which are distinguished from beneficial symbionts, and also transients as well, which I think is a really nice, so it's the right column thing to think about as well, a bit more, like maybe the food that these animals are eating, microbes that are on the leaves, for example, might come through. And while they may not actually influence the host anyway, they're just kind of along for the ride. So we should think about these different things when we're studying host-microbe interactions, is what they're trying to say here. They then surveyed the literature for evidence for groups of animals for where there's kind of little or no evidence for there being beneficial symbionts. And so there's a few groups of animals here. One thing to note is they're not saying that none of these groups of animals have beneficial microbes, right? So, there's ants here, right? And, Veronica, we know there's a lot of really beneficial ants for some species, microbes for certain species of ants. But what they're saying is that they've also found evidence for some ant species that don't have beneficial microbes. So we shouldn't just assume that all ants have microbes. We should really be kind of careful and, think of it in that way. They also, back themselves up a bit, which is nice. They say, while it is impossible to disprove the existence of undetected microbes or microbial functions in any of the animals mentioned above, we argue that the microbiome-free state is the appropriate null hypothesis and that evidence must be acquired to reject it. So they're basically saying, we shouldn't just assume everything has microbes. We should really, for some of these groups, really try to do our due diligence to really confirm that. They also talk a bit about low microbiome animals. And so, people here may have studied a few different taxa that may have kind of a low microbial biomass. So maybe some, fish, for example, there's some evidence for. Birds and bats as well, people have found that, there's maybe not a huge amount of microbes there. They also say, their microbiomes are dominated by kind of weedy kind of taxa that aren't, ones we don't commonly find in like, herbivores, etc. They also kind of point out terrestrial carnivores. And ultimately, they kind of summarize that by saying, some species within these groups have relatively simple guts with short retention times, traits that also suggest a lower reliance on microbes for digestion and nutrition. And this kind of makes sense, right? So if you're like a cattle, you're eating plants, which are very hard to get a lot of energy from. A lot of that is like locked away into these complex, cell wall compounds. You need a rich community of microbes to break that down, ferment it, so that you can get energy. But for some animals, so bats, birds that maybe they eat fruit or other proteinaceous things like insects and stuff. A lot of those things are probably quite high in bioavailability, right? They don't need a secondary kind of metabolism to actually get energy from that. So yeah, they're essentially saying, think a bit about the natural history of the animal as well that you're looking at. Don't just assume that it has a microbiome. Think a bit more about that. And then also talk a bit about, why do we actually think that all animals have microbiomes? Why is that kind of like the notion in the field? I mean, one thing is that we've studied animals, we study humans and mice a lot. That's where a lot of our understanding comes from. They also mention tools used for the characterization of microbiomes are getting cheaper and cheaper and easier to use, right? So 16S sequencing, for example, is very simple, easy to use. And we don't have to do experiments to actually test whether that has an impact on the animals at all. You don't necessarily need specialized knowledge on your host species. So if you're really just interested in having a look, you don't really have to be a physiologist or understand the natural history of that animal very well. And then they talk about contaminants and transients can also look a lot like a beneficial microbiome if you don't control for them. And so I'm going to talk a little bit briefly about these two things, contaminants and transients, and what we know about them and how you can kind of control for them, etc. So what do I mean by contaminants? So contaminants are essentially DNA or cells that are exogenous, that is, they come from outside of the sample that you're studying. So this can be, for example, skin or environmental microbes that come into your tube when you're collecting your sample in the field. It could be from laboratory reagents and kits. So we actually know that like certain kits have certain contaminant taxa in them. Some people call it the kitome, which is a funny phrase. The laboratory technician again, so you could be oral microbes or skin microbes, or even from the lab environment. I think there's some evidence that some microbes, we've actually been selecting certain microbes to grow in lab spaces. Some can degrade alcohol and stuff like that. So we're probably breeding some really nasty superbugs in labs. And so they can also get into your samples and contaminate your results. A key thing to note here, though, is that DNA contamination affects samples with lower input biomass more severely, right? So this kind of makes sense. If you have a sample that has very few bacterial cells in it, and you try to study the microbiome of that, contamination, which is usually at a kind of low level relative, is going to be much more significant, right? Whereas if you have like a soil sample, which we know is really rich in bacterial DNA, any small amount of contamination isn't really going to affect that because the endogenous DNA is going to kind of outcompete that when you're processing it and analyzing it. So a little bit of background. So I actually did my PhD in the field of ancient DNA and actually dental tartar, so like studying the microbes in dental tartar. And people who know a bit about the ancient DNA field will know that like contamination has been a really big kind of thing historically. So there were some studies from the 90s and such that made these really bold claims of isolating microbes from amber or dinosaurs. That's been met with skepticism as well. I think it's safe to say most people think that that's probably not the case. You probably can't get DNA from dinosaurs or amber. But it's still it's a bit of a shame. But there has been a bit of contamination is something in the ancient DNA field that people have known about for a long time, right? And people have really tried to create a framework to counteract that and control for that. Even in my PhD, which was from 2015 onwards, there was still a lot of kind of dodgy stuff going on. I actually wrote 3 replies to papers that kind of I thought didn't really authenticate the results properly. So yeah, there's still some dodgy stuff going on. But what about the modern? Not everyone studies ancient DNA. What about modern microbiome research? Well, this is the placenta. Have people heard about the placenta microbiome at all? This is like another very kind of contentious topic in the field, right? And I think it kind of roughly originated from this paper here in 2014, where some clinicians basically investigated the placenta to see whether it had a microbiome or not, using 16S amplicon sequencing. And they didn't really control very well. They didn't control the contaminants at all very well in the study. As a result, there's been a whole bunch of studies kind of refuting the idea of a placenta microbiome. People have tried to replicate these results using big data sets, using adequate controls. And really, I think the general consensus now is that there's probably not a microbiome in the placenta, that it's probably the result of contamination. So it's nice that, science can kind of come around and correct things, but, this is still a problem. It's not just the placenta as well. It also affects a whole bunch of other body sites. This paper here is a real, seminal paper for contamination and microbiome research by Susanna Salter in 2014, where they really, bring this idea up that, reagent contamination and laboratory contamination can really impact your microbiome analyses if you don't control for it. So during my PhD as well, I saw there was still a lot of, these kind of dodgy studies going on, things like the brain microbiome, I know the penis microbiome, whatever. And so I rather than kind of going after each of these separately, I thought, well, why not actually create like more of a framework that reviewers, authors, editors can look at and say, is there a checklist that people can use? Have they actually properly controlled for their microbiome study for contamination? And so we published a study here. So if you want to learn a bit more about contamination, some of the history and how to control for it, definitely recommend a couple of these papers. So it's contamination. Onto transients now, which can also impact our interpretation of microbiome studies. The quote that they use in the Hammer paper is, for example, 2 caterpillars may appear to have different microbiomes simply because they consume leaves with different types and amounts of microbes, even if these microbes only pass through the gut. So I think this is actually based on the study that Hammer did, where they found that, it looks like transients were driving these, signatures that you could have interpreted as different beneficial microbiomes. Another thing that I would add as well is that we really also have to be careful about DNA not equal living, right? When we're sequencing DNA, we're not actually, we don't know if it's living or dead. So that's another thing we should also think about a bit more when we're studying these things. Figure 2 is like a nice little overview of signs that an animal may lack a microbiome. So the first one is talking at, like if you sequence some controls, maybe that's the diet, it's the lab reagents and stuff like that. If most of your data is coming from microbes that you find in these kind of controls, then maybe you don't have much of a microbiome, or it's a very low microbiome animal. There's also some other great tools you can use. So qPCR, so actually measuring the quantity of DNA in a sample is really powerful. So you can compare, say, your target animal to a reference animal, or also I would recommend against negative controls in your study. And so you can determine like a limit of detection for whether there's actually microbes there or not. And then also microscopy is also a really fantastic way as well. So there's some great studies, people, using fluorescent microscopy to look at whether microbes are actually in the guts of certain animals. And that's another nice line of evidence you can use. The last one, this one here is more about the physiology of the animal. So does the animal have behaviors for transmitting symbionts between generations? That's a really important thing we have to think about. Do they have a very fast transit time through the gut? Is it a very simple gut? Do they have any specialized symbiont housing organs? And so if the answer is no to some of these, then you might want to be a bit more careful with your study species. And the last one is about function. But again, this is obviously, it's very hard to do functional kind of studies like experiments with animals where you so for a lot of animals, this is very hard to do, but they're saying, obviously you can test the functions. Are these microbes important for the survival of these animals? Which we would love to do, obviously, but it's hard. And the quote that I like here is, it's important to not just ask who are the microbes and what are they doing, but also whether there is even a microbiome in the first place. So keep this in mind. I'll kind of finish with this kind of summary quote by this paper, which I thought was really cool. So by generalizing all animals as microbe-rich holobionts, this paradigm obscures an interesting aspect of animal diversity, that animals vary strongly in the degree to which they host and depend on microbial symbionts. This more nuanced perspective encourages us to rethink the approaches we use to study animal microbiomes and prompts new questions about how and why animals evolve along a spectrum of microbiome dependence. And so this is pretty cool, right? So this raises a lot of really interesting evolutionary questions, like why do some animals rely on microbes? Why do they outsource the degradation of plant compounds to microbes, whereas some have them encoded by their genomes? Bioluminescence, so there's like a squid that has bacterial symbionts that produce bioluminescence. They do that, but why do some other marine organisms do it by themselves with their own genomes? I think these are really interesting questions as well. And so, that's really cool. Cool. So that's kind of like a summary of the Hammer paper. And now I'll talk a little bit about the question, which I think is related. It's like, how much of my metagenome is of microbial origin? Which seems like a very simple question, right? But it's actually a bit more nuanced and difficult to answer. So one way you could do this, right, is you could filter out all the DNA in a sample that's not microbial in origin. So what we normally do is we filter out the host DNA. So we map our reads to like the host genome. So that could be the human genome, or if you're studying a human sample. The problem there, right, is what if you have an organism that doesn't have a good reference genome? You know, we're studying a novel species that doesn't have a host reference genome yet. It's really hard to filter that. What about dietary DNA? So if you're studying a fecal sample, what if there's a lot of prey items, so insects or other vertebrates, and plant DNA? How do you get rid of that? Well, there's no chance in hell you can do that because it's very difficult. We don't have the entire genomic catalog of organisms to actually do this. So getting rid of the DNA that's not microbial is kind of intractable at this point. So why I got into this was from a study that I'm working on currently here on 3 different species of bats. And so this is studying the fecal microbiomes, 3 species of bats that have never been studied before. So we went through, we did our metagenomic sequencing, we did our genome-resolved metagenomics pipeline. So that's creating genomes from the metagenomes. So the first thing I did was map these fecal samples to the host genomes. We didn't actually have perfect reference genomes as well, sometimes only genus level representatives, so that's not perfect. And we still get quite a bit of DNA that's not host, so we can actually still work with this data set, which is great. I then went to do the assembly, the binning, and mapping. So we basically create these genomes from the metagenomes, and then we map the metagenomic reads back to those reference genomes. And that kind of lets us know what kind of microbes are there. And we got a really low proportion of reads mapping to these 3 different species of bats. So we're like, that's interesting. I would have expected maybe more, right? Like typically when you do a human study, you get 80% plus of your reads mapping to the bacterial genomes in your sample. So there's kind of 2 things that could be causing this, right? So one is we haven't sequenced deeply enough. And the other is that maybe there's actually a low amount of bacterial and archaeal DNA in these samples. So for the first one, right, when we're doing our genome assemblies, like the coverage is really important, right? So say we have this kind of like blue genome in our sample that we're trying to assemble, and the green is the reads that we kind of create, and we try to join them up and reconstruct this genome. In situations where you don't have enough sequencing coverage, it's obviously going to be impossible to kind of create this genome assembly. And it can also be very difficult in very diverse samples, right? So if you have a very high diversity of microbes in a sample, then obviously you need a lot more sequencing reads to cover all the genomes in that sample. So therefore, that could be one of the issues that's causing this issue. So to test the kind of first idea that maybe we haven't sequenced deeply enough, we just threw more money at it. We just sequenced it twice as deeply. And what happens? Nothing. No. Same. Same amount of reads mapping to the mag catalogs. So obviously, it seems like this isn't the case, right? Maybe the diversity was saturated, but still really demonstrating that the low amount of these reads that are mapping to these genomes is because there's actually a low amount of bacterial DNA. It's very difficult. Like, well, at least I couldn't find a way in the literature that lets you do this and kind of be sure about it. So I was thinking about it a bit more. There's a very nice characteristic of bacterial genomes that they have these single copy core genes. And so these are genes that have very conserved functions. They're kind of universal across bacteria, which is great. And they're usually always found in one single copy. So people use these for various things like assigning taxonomy, etc. But it's a nice feature that we can use maybe to look into this problem. So I thought rather than try to filter out all the non-bacterial DNA, why don't we just estimate what proportion of it is actually bacteria? Because they have very beautiful genome structures that we can exploit for this. So before I went and tried to write some code to do this, to estimate the single copy core genes, I looked around first because you don't want to reinvent the wheel if you don't have to. You don't have to see if there's actually a tool already around. The closest thing I could find was Single M, which is developed by Ben Woodcroft from Queensland in Australia. And this is a really great pipeline he's developed for characterizing de novo OTUs from single copy core genes and metagenomes. So it's pretty much what I wanted to get those kind of counts of the single copy core genes. It's very cool. It's reference agnostic. So you don't need reference genomes. You don't have to know what's in your sample when you use this. It doesn't require the assembly, right? You can do this kind of on the reads that you come off the sequencing machine. So it's quite nice in that sense. It doesn't require really computationally heavy tasks. And it's also very fast and scalable, which I'll show a bit later. So I got in touch with Ben and asked about this. We had a chat about this, and he said, yeah, I think it could probably work. So we worked together and created some code to estimate the amount of bacterial and archaeal base pairs in your metagenome. And so essentially how I do this, I'll give you a brief rundown of how this works. So what I did is I calculated the mean genome sizes for each taxonomic rank in the genome taxonomy database. And then the kind of calculation for how much of your metagenome or how much of a bacterial's genome, of metagenome is bacterial, is basically the mean coverage of the single copy core genes multiplied by the mean genome size in the lowest rank. So I'll give you an example of this. So say we have single copy core genes in our sample that belong to E. coli, and these single copy core genes are covered 5-fold. We assume that the genome, therefore, of E. coli is covered 5-fold, and we just get the mean genome size of E. coli and multiply that by the coverage. So that's like 5 megabase pairs times 5 coverage, 25 megabase pairs in our metagenome. I did a bunch of simulations on simulated metagenomes to demonstrate the accuracy of the method. I'm not going to show you that, but if you want, you can come and look at my poster, which is not hanging at the moment, but I could show you anyway. Another cool thing, so this is showing the scalability of this approach. So Ben ran single M on 140,000 metagenomes from the SRA, which is pretty cool. And then we estimated the bacterial and archaeal fractions. And this is like a density plot for different metadata categories of samples. And so the middle top one there is the human gut metagenome. As you can see, it's the bulk of the data in the SRA is human gut microbiomes, and that's like 35,000 samples. You can see the density. So on the x-axis here is the estimated bacterial archaeal percentage of the metagenome, and that's about 80% plus, right? So we kind of know this, right? Human fecal samples have a lot of bacterial and archaeal DNA. Soil as well is really high. But then if we start looking at these other body sites, right, like the human oral metagenome, the plant metagenome, marine metagenome, and food metagenome, it's a much more different story. It's a bit more nuanced. It's not just like this huge amount of bacterial archaeal DNA. Some of these really have bugger all bacterial DNA in them at all. Okay, so anyway, we developed a tool that could hopefully answer the issue that I've been having with the bat stuff. So let's go back to the bats. So this is plotting the percentage of reads mapping to the MAG catalog, the genome catalog we created, versus the single M estimates of the bacterial and archaeal fraction. And there's a really nice correlation between these two, which is fantastic. And this is showing this data again, but like in a different way. This is just grouped by the different bat species. And this is the single M bacterial archaeal estimates of the metagenomes. What's kind of cool is you can see that the different species of bats have different kind of average proportions of bacterial and archaeal DNA, which is interesting. So maybe we need to treat them differently when we do our analyses subsequently. But if we then just compare these back to the average read counts to the MAG catalog, you can see they're actually very close to each other, right? So 24% to 24%, etc. Which is, to me, this is awesome. I was like, yes, it's like, looks like this method is actually working, which is fantastic. And what this is therefore led us to say is we've actually exhausted the diversity of the bacteria and archaea in our samples. So we don't have to see what's any deeper. We can actually also demonstrate that this is actually a low microbiome animal in this regard, or these samples are low microbiome, which is nice. So anyway, concluding, I hoped I, convinced you a little bit about why we shouldn't assume that all animals have rich and diverse microbiomes. And I've also showed you that we've developed a tool that can kind of help in this regards, particularly with genome-resolved metagenomics, which a lot of people are using here in this group. This can save you money, as you can see from our example. And also, I think it will help us in general to, as we explore the microbiomes of more diverse host species, right? We're moving beyond just humans, mice, etc. Some of the stuff we're doing in the EHI, etc. Bat scats don't seem to have a lot of bacterial and archaeal DNA, which is kind of interesting. And I think, again, I hope I've convinced you that we shouldn't assume that metagenomes are always really rich and dense in microbes. So, yeah, thanks.